Welcome to the Alamo Breast Cancer Foundation Advocate Chat. I am Dale Eastman, member of the Alamo Breast Cancer Foundation Advocate Committee at the San Antonio Breast Cancer Symposium. I serve as Chair of Advocacy and Government Relations for the Alamo Breast Cancer Foundation. I am a 28-year breast cancer survivor and a 3-year thriver of metastatic breast cancer. Recently, there has been a lot of news shared among breast cancer organizations and other organizations regarding a blood test that will be able to screen for multiple cancers. The Advocate Chat today will help demystify information we have been receiving. I am delighted to introduce you to Megan Hall, Ph.D., Senior Director of Medical Communications for Grail, a company that has a multi-cancer early detection test in development. During the chat, I will be referring to multi-cancer early detection also as MCED, which is the initials for multi-cancer early detection. So welcome, Dr. Hall. We appreciate you being here today.
Thank you so much for the opportunity.
So we will start with the questions. What exactly is a multi-cancer early detection blood test, MCED?
So I think to understand what a multi-cancer early detection test is, it's important to understand the current state of cancer screening. Right now, there are recommended screening tests for only 5 different types of cancers: breast cancer, colorectal cancer, prostate cancer, cervical cancer, and in high-risk patients, lung cancer. And what that means is that the vast majority of cancer types have no recommended screening test at all. And so patients tend to appear at their physician when they have symptoms. And when you have symptoms, that cancer tends to be much later stage, is much harder to treat, and outcomes tend to be very poor. So a multi-cancer early detection test is a single blood test that can detect almost any kind of cancer from a single blood draw. And importantly, the Gallery test, our test, can tell you where in the body, or predict where in the body, that cancer signal is coming from. So it's a really significant advance in cancer screening, and we think a way that we can bend the cancer mortality curve by detecting many more cancers at earlier stages.
So is MCED considered like a liquid biopsy?
Liquid biopsy has traditionally been used to discuss tests or to address tests that are used in patients who are already diagnosed with cancer. So most people might be familiar with tests that will tell you if you have an actionable mutation. So for example, in lung cancer, looking for an EGFR mutation once you're already diagnosed and you're comparing to the tumor, right? So this test is actually in patients who are asymptomatic and have not yet been diagnosed for cancer. And so we've been very careful to not call it a liquid biopsy specifically, because it's technically not doing the same thing that the liquid biopsy tests are doing. It's really meant for early detection and screening, hence the moniker multi-cancer early detection test.
Well, thank you for clarifying that. Several companies are also developing multi-cancer early detection screening. Will there be a difference between these blood tests?
So there are a few key differences, I think, in the blood tests. And first, I want to say all of these tests are generally based on what's called circulating cell-free DNA. So they're detecting tumor DNA in the bloodstream. Some of the tests are using mutations, so point mutations that appear in the tumor. Our test, the Gallery test, is using methylation patterns. So there are a number of different kinds of genomic alterations, or alterations in the DNA that can be present in the tumor that then get shed into the bloodstream, and these tests are picking up those genetic alterations. We have looked at various kinds of genetic alterations. We've looked at mutations. We've looked at larger copy number alterations, so bigger insertions and deletions in the chromosomes. And we've also looked at methylation patterns, which are methyl molecules attached to the DNA itself. And what we found was that methylation actually outperformed the other two genomic alterations when we were looking for cancer detection, as well as the ability to predict where in the body that cancer signal was coming from. So that's one key differentiator between the tests, is the underlying technology that they're using. The other is that our test includes this cancer signal origin prediction capability. And that's really important because it's meant to direct the downstream diagnostic workup. So instead of defaulting to something like a whole body scan, a whole body PET CT, this can really direct the physician and the patient in determining what targeted diagnostic tests to do in following a positive result.
Okay, thank you. MCD is going to screen for more than 50 cancers, which I think is just totally amazing. Are there any cancers that will not be able to be detected on the blood test?
So you're hitting on a really interesting biological driver of the test. So I've mentioned that these multi-cancer early detection tests are detecting cell-free DNA fragments in the blood, and those fragments carry cancer-specific genomic alterations. Interestingly, the more aggressive cancer types tend to shed more DNA into the bloodstream. That makes them inherently more detectable by these assays. The flip side of that is that cancers that are less aggressive or tend to be more indolent do not shed as much DNA into the bloodstream, and that makes them inherently less detectable by the assay. Those cancer types include early stage prostate cancer, thyroid cancer, both of which we know tend to be subject to some overdiagnosis and overtreatment. So I always say, for example, if we detected every single stage 1 prostate cancer, I would be very worried about the test. But that's not what we're seeing, because the biological driver of detection is this shed rate and how much cell-free DNA is present in the system. And it really means that we are detecting cancers that are more aggressive, really should be getting treatment, and we're not subject, at least in our data, to things like overdiagnosis and ultimately overtreatment.
Okay. So most cancers have no screening options. However, we know that breast, cervical, colon, and prostate, and what you said also types of lung cancer, have routine care cancer screening tests. Is MCED designed to be complementary to those cancers that have existing routine tests?
Absolutely. MCED tests are absolutely meant to complement, not replace, the existing recommended screening tests. These recommended screening tests, mammography, for example, colonoscopy, stool-based testing, these are really important tests on a population scale. We know there are benefits. We don't want to miss out on those benefits. Importantly, those tests are designed differently than multi-cancer early detection tests. And those tests are designed to tolerate a higher false positive rate than a multi-cancer early detection test. And they can tolerate a higher false positive rate because the workup is really organ directed. You know exactly where to look for the cancer. In a multi-cancer early detection test, for example, without the ability to predict the CSO, the specificity or false positive rate becomes really, really critical. And so these tests have been optimized for false positive rate to keep that as low as possible, to really, the last thing we want to do is shunt a lot of asymptomatic patients unnecessarily into the healthcare system for false positive workups, for example. So the false positive rate for our test is less than 1%. Right now it's about 0.5%. And what this translates to is a positive predictive value, and this is the chance that if you get a positive test, that test is accurate, of about 45%, which is important to put into context with the existing recommended screens. Again, these tolerate a higher false positive rate because they are very organ specific. So if you look at the PPV for something like mammography, that PPV, or positive predictive value, is about 4.5%. So that means between 4 and 5 out of every 100 positive tests is actually a cancer. Because we're screening for so many different cancer types, our PPV is much higher because we're capturing so many different cancer types.
So you are promoting continuing routine screening.
Absolutely, absolutely. Patients really should be working with their healthcare provider to ensure that they are getting their mammograms, getting your colonoscopies or your stool-based testing. Any screening is better than no screening. If you're a high-risk patient, get your LDCT for lung cancer. Work with your physician to determine if you're appropriate for prostate cancer screening. And of course, women should be getting cervical cancer screening. In addition to, now that we have the Gallery test, we can layer that on top.
Okay. Should MCED become incorporated into routine medical care without discouraging patients from routine screening?
Absolutely. I think that would be the ideal state, would be where a patient could, for example, go in for an annual exam. Patients often are having blood drawn anyway to check, triglycerides, looking at your cholesterol. Could be relatively simple to implement an additional blood draw, a couple of tubes for the Gallery test on that same time frame. And so this, in addition to the existing recommended screening tests, should significantly increase the number of cancers that we detect and should significantly decrease the population mortality that we are seeing from cancer.
So you were talking about the blood testing. How many vials of blood are needed for this test? I'm just curious.
It's 2 tubes of blood, 2 10 ml tubes of blood, which is a pretty standard amount for a blood test.
Okay. Do you expect at least a 5-year cancer survival benefit by finding a cancer in a blood test before symptoms appear?
We do. We know right now that earlier detection is associated with improved survival. So, for example, the 5-year cancer-specific survival, when a cancer is diagnosed at a late stage, when the cancer has already spread, is only 2 in 10. However, when you diagnose a cancer at an earlier stage, so before the cancer is spread, that 5-year cancer-specific survival increases to almost 90%. So we know that there's a benefit to earlier detection and to detecting cancers at earlier stages before the cancer is spread. And that's exactly what we expect to see when we implement the Gallery test, as we are right now, on a population scale.
Okay. Can you describe the science behind MCED? Since this is going to be at the San Antonio Breast Cancer Symposium and advocates and researchers and clinicians are going to be watching this, could you address the science?
Absolutely.
Okay.
So and I'll back up a little bit before I get into the technicalities of the way the Gallery test works, and I'll just talk briefly about how this technology was discovered and how Grail was born. So there's another cell-free DNA-based technology called non-invasive prenatal testing, and this is a technology that is looking for fetal cell-free DNA in the mother's circulation. And Illumina was implementing this on a large scale in hundreds of thousands of pregnant women. And what they found was this anomalous signal in otherwise healthy pregnancies. So the fetus was fine, but the signal was aberrant, was off. And what that signal was representing was undiagnosed cancer in the mother. And so the idea that we can leverage cell-free DNA for cancer screening for multiple types of cancer was born, and Grail was initiated. And so in the past 5+ years, we've now come to this place where we have Gallery on the market, and we're leveraging the cell-free DNA that was originally detected from this non-invasive prenatal testing in our technology. The way that our technology works, so cancers carry genomic alterations, of course. They have mutations, methylation pattern differences, copy number differences, so larger genomic insertions and deletions. Cancers are also, as they're turning over, are shedding DNA into the blood, and that DNA in the blood is carrying those cancer-specific markers. So our technology is able to detect from the blood cell-free DNA that is cancer-specific and carrying those cancer-specific markers. So we use methylation patterns specifically, and methylation is a driver of cell differentiation. So it's very important in determining how a skin cell becomes a skin cell, how a liver cell becomes a liver cell, for example. But it's also very important in cancer generation. And so we're leveraging that biological signal to do cancer signal detection. It's very specific for differentiating a cancer from a non-cancer signal. And then also for the cancer signal origin prediction, because it's so specific to unique tissues and cancer types, we're able to leverage that methylation signal from the DNA fragments that are present in the blood to predict a cancer signal origin in the body.
Could you just define methylation?
Sure. So methylation is what we call an epigenetic alteration. It's not an alteration in the actual DNA sequence itself, but it's a molecule that is added to the DNA that generally affects how genes are expressed. And so what genes are turned on and what genes are turned off. And what that does is really drive tissue differentiation, as we've mentioned already. It also can drive cancer cell generation and tumor growth. And so these epigenetic alterations are pervasive across the genome. There are 30 million different, we call them CPG markers. So it's a methyl group attached to a specific sequence in the DNA. There's about 30 million of those across the genome. And our technology is looking at 1 million of those CPGs to look for very specific cancer signals and then also to determine the cancer signal origin itself.
So talking about the clinical trials, can you divulge the positive clinical evidence resulting from the MCED studies?
Absolutely. So our foundational study is called the Circulating Cell-Free Genome Atlas Study. This is a case control study where we very carefully developed and validated the performance of the multi-cancer early detection test, Gallery. And we recently published a cohort from that study that was over 4,000 participants, clinically validating the performance of the test. And that's where we found a less than 1% false positive rate, so a false positive rate of 0.5%. We have a cancer signal origin accuracy of over 90% for the top two predictions. And then we have a positive predictive value, a modeled positive predictive value of 44%. And I say that's modeled because a case control study is, of course, going to be artificially enriched in cancer cases, which is not going to represent the cancer stage and type distribution in a population. And so we would overestimate the PPV if we didn't back adjust that for what we expect to see in the population. And so that's why I say it's a modeled PPV from our case control study, because we adjusted that based on what we expect to see in the population using the expected SEER distributions. Importantly, we have an interventional study called Pathfinder that was returning results to clinicians and patients so that we could understand clinical implementation of the test and also understand performance in the real world. And what we saw there was a very consistent positive predictive value. So the positive predictive value was between 40 and 45% when we implement this in a real world population. So this suggests that our test has been developed extremely rigorously. The performance is translating as expected. And so we expect to see that performance carry over to any number of different patient populations. We do have a number of clinical validation studies that are also ongoing. Overall, with our 4 foundational studies, that's CCGA that I already mentioned, Pathfinder, we have a study called Strive, that's 100,000 women undergoing their screening mammography. We have another study called Summit that is in high-risk patients due to lung cancer. Overall, that clinical program is going to be more than 145,000 participants. And then, of course, we have our partnership with the NHS in the UK. That study on its own that's a randomized controlled trial that's going to enroll over 160,000 participants. So overall, our clinical program is going to be over 300,000 participants, and it's growing as we develop new studies. So it's one of the most robust clinical evidence programs that I've ever seen in genomic medicine, and I'm constantly impressed with the data that's coming out of our teams.
I agree, that's quite impressive. You mentioned false positive, but were there other negatives that might have come out of the studies that have already been out there?
So there is always the potential for a false negative as well, and that is partially due to what we had talked about before, which is the biology of the signal, that more aggressive tumors are going to be detected better than less aggressive tumors. And so, for example, the test will not detect every single early stage prostate cancer, but that's also not necessarily a terrible performance characteristic because it means that we're not going to be detecting the more indolent cancers that might be subject to overdiagnosis and overtreatment.
So you are going to have studies now that are going to be enrolling up to 300,000. That's wonderful. But how many individuals participated so far in the studies?
So we have enrolled to date over 120,000 participants. So our Strive study is fully enrolled, Summit is fully enrolled, CCGA is fully enrolled, and I think it's actually upwards of 120,000 so far. The NHS Gallery study in the UK is actively enrolling very quickly as well. So these numbers are going to go up very fast.
So do you have a lot of United States participants in the studies?
We do, yes. So the core set of foundational studies is largely based in the US. So CCGA, for example, we partnered with 142 institutions across the United States and one in Canada. And so we wanted a very broad representation of participants in the study. And we've been able to partner with some pretty high profile leading institutions like Mayo, Dana-Farber, and Cleveland Clinic, to name just a few.
Sure, that's good. Were there people of different gender and ethnicities?
Absolutely, yes. So different genders, yes. CCGA enrolled men and women. Most of the studies are enrolling men and women. The only one that we have right now that is focused on women specifically is the Strive study, which was targeting women undergoing their screening mammography. So that one, of course, is restricted to women. But the remaining studies are men and women. We are very actively engaged in making sure that we have good racial and ethnic diversity in our studies. I think this is a challenge that we need to be able to address. We're not where we would like to be right now, but we have programs that we are ramping up to ensure that we have good racial and ethnic diversity in our studies as much as we can moving forward.
Okay, that's good. What were the ages of the individuals in the studies?
So CCGA was almost all ages, adults, any adult age. So we have patients as young as 21, 22 in the CCGA study, but not very many. The remaining studies tend to be restricted to older patients where we know that the cancer risk is greater. And so Pathfinder, for example, is aged 50 and above. And that's also going to be the case in a fair bit of our other studies. And that's largely because we know that age is unfortunately one of the greatest risk factors for cancer. So, for example, a patient who's 50 years old has about a 13 times greater risk of having any cancer than the general population. And so it makes sense when you're looking at a cancer screening technology to look in the population where there's a little bit of an elevated risk.
So what would you say would be the suggested age to begin an MCED test?
Right now, Gallery is recommended for patients who are what we call at elevated risk of cancer, for example, age 50 and above. There are potentially other risk factors that could also make a patient appropriate for Gallery. This could include genetic predisposition, family history, or other comorbidities. And that's a discussion that we encourage patients to have with their healthcare provider to determine what that elevated risk profile might look like for them.
Okay. Okay. Well, let's touch a little bit on breast cancer. Can women or men who receive a MCED test not only find out that they have breast cancer, but also may have an additional cancer or cancers present in the blood specimen?
So technically, yes, that would be possible. I think that is going to be extremely rare. The cancer incidence is relatively low in the general population, but the technology is such that we can detect signal from multiple cancer types. And so it is possible that we would detect a patient who has breast and another type of cancer.
So, addressing breast cancer again, can finding breast cancer in blood also, and I think you touched on this and I find that amazing, also provide information on the location of the breast cancer?
The location would, yes. So the location or the cancer signal origin prediction on the test would say, if it is predicting a breast cancer, it would say cancer signal prediction breast. And so it is able to predict one of 21 different anatomic locations, anatomic or histologic locations for the cancer. So breast cancer is one of those CSO predictions.
So it can actually point out metastatic breast cancer.
So what the test does not do is tell you the stage of the cancer. That would have to be determined upon diagnostic workup and confirmation with the healthcare provider and potentially pathologists. What the test will do is predict where the signal origin is coming from, and that can very specifically direct that downstream diagnostic workup. So, for example, a patient who has a cancer signal detected and a cancer signal origin prediction of breast cancer could potentially, and we've seen this from our data, could potentially go in for a diagnostic mammogram to definitively determine the presence of cancer, and then ultimately biopsies can help determine stage.
So finding breast cancer in the blood does suggest further screening and diagnostics are going to be needed.
Definitely, yes, absolutely. And so this is the first step to getting to that definitive diagnosis. And it's, again, meant for asymptomatic patients. And so this is really where the importance of the cancer signal origin prediction capability comes in, because we want to be able to direct that downstream diagnostic workup to focus on a specific area to avoid a diagnostic odyssey.
Is $949.99 the suggested cost for a multi-cancer early detection blood test?
That right now is the list price for Gallery, yes. But we do understand very keenly the importance of access, and coverage is going to be part of that access. And so we have teams working with payer groups on getting this test covered as soon as we can.
So where does MCED test stand on getting insurance coverage?
Well, as I said, we definitely recognize the importance of having this test accessible as broadly as possible, as soon as possible. And part of that will be having this covered by insurance, by Medicare. And so we have teams working on this. I can't really speak to a timeline at this stage, but know that it is very important to us because we really want to ensure that we can address the health inequalities that are in existence, particularly with respect to cancer screening and care.
Well, the good news is that MCED Medicare coverage has been reintroduced in the U.S. 117th 2021 Congress. And many cancer organizations, including the Alamo Breast Cancer Foundation, are supporting the passage of House Bill 1946 and Senate Bill 1873. What is the status at the present time of the movement of these bills?
Well, we absolutely stand with your organization and all of the other advocacy and political organizations who have supported this legislature. This is really critical for coverage and, as we have discussed already, for access. And so we are very supportive of these efforts and are very optimistic that these will both pass so that we can have coverage of this kind of important technology, because we know that earlier detection saves lives.
And I think with passage of these bills, that will help with getting the insurance coverage that will be needed too. Do you predict the MCED blood test will receive FDA approval by 2023?
Well, it's hard to speak to timelines specifically, but what I can say is that our technology has received breakthrough device designation from the FDA. So the FDA recognizes the importance of this technology, and we are working very closely with the regulatory agency to ensure that we can get FDA approval, because that will feed into a lot of other things that will make this test very accessible. It can help insurance coverage, it can help guideline incorporation. All of these things are really important for ensuring broad access of this technology.
Well, this concludes the chat, and we thank you, Dr. Hall, for your participation in the ABCF Advocate Chat and providing us with the information about the multi-cancer early detection screening blood test. I hope that the future of MCED will prove to detect cancers early enough that outcomes are going to improve. Maybe sometime in the future, we can have another chat for an update on the results and addressing access, affordability, and insurance coverage.
I would really value that opportunity. Thank you so much for your time today. I really appreciate it.